Good afternoon. Welcome back to virology. So the last two sessions we talked about making RNA from RNA and from DNA. And so today the last process, making DNA from DNA. And of course, just like RNA genomes, DNA genomes need to be replicated to make new progeny. And so the DNA genomes, of course, include double-stranded DNA, single-stranded DNA, and gapped double-stranded DNA. So that's what we're going to talk about today. And remember, I think you know this already, but good to emphasize, DNA replication is always delayed after infection because you need to make some proteins. So at least one, because as you'll see today, if the virus needs to use the host DNA replication system, it needs to make one protein that will attract that system to the viral genome. And if you have viruses with big genomes that encode DNA polymerase, those proteins have to be made first. So that's why DNA synthesis is always delayed after infection. So just like for RNA, there are universal rules of DNA replication. Here on the top left is our schematic of the template and the primer. So when a primer is used, the 3' hydroxyl is used to add new bases that are complementary to the template. And the template is read in a 3' to 5' direction. The new bases are added in a 5' to 3' direction. DNA replication is semi-conservative. So up in the upper right there, this shows you what semi-conservative is. If you start with a double-stranded DNA here, it's colored as RNA. I don't know why, but it's the same rules apply for RNA. Double-stranded molecule, each strand is copied. So the two strands separate and they're each copied. That's semi-conservative. There are some cases where only one strand is copied. That's conservative. I don't know why they're called semi-conservative and conservative, but that's the nomenclature. The place where replication begins is called an origin. We're going to talk a lot about origins today, origins of replication. DNA synthesis is catalyzed by DNA-dependent DNA polymerase and accessory proteins. And DNA synthesis can be primer-dependent or primer-independent. In fact, when I was teaching this course more than, 5 years ago, I always used to say DNA synthesis was primer-dependent always. But you have to be careful about making always statements in biology. This experiment overturned this dogma. This is a DNA polymerase assay where on the y-axis we're looking at incorporation of TMP into product, and on the x-axis is time. And so T7 DNA polymerase is isolated from a bacteriophage. This is primer-dependent. If you do the reaction without a primer, that's the orange circles, you don't get any synthesis of DNA. If you add a primer, that's the red, then you get nice synthesis of DNA. But here is a DNA polymerase encoded in the genome of a bacteriophage called NRS1. This phage was isolated from bacteria around the deep sea vents. You know those things at the bottom of the ocean that are bubbling and smoking? There are lots of life living around it, including bacteria. And when there's life, there are viruses. So this virus was isolated from one of those deep dwelling bacteria. And look, NRS1 without a primer is making product as much as with a primer. So dogma overturned. Some presumably more DNA polymerases are primer independent that we haven't found. The synthesis of DNA goes by a two-metal process, just like RNA. On the left is the structure of a DNA polymerase at the top there. And on the bottom is the poliovirus RNA-dependent RNA polymerase. Just to remind you that they're right hands, where the palm is the active domain. The polio polymerase, the thumb and fingers touch, so there's an enclosed active site. But for the DNA polymerase, it's open. The thumb and four fingers are not closed. So there's the active site. And the two yellow are the beta strands that contain the aspartates that coordinate the magnesium metals. And just like for RNA, you have a template, you have a series of riboses joined by phosphates with bases coming from them. And here we're showing the addition of a base, a T complementary to A. The T is brought into the polymerase as a TTP, 3 phosphates. And then the magnesium ions, metals that are held in place by the aspartate help carry out these reactions where the last 2 phosphates are released. The energy from that goes to making the final bond of the phosphate to the ribose. So you end up with 1 phosphate between the 2 riboses, very much like RNA. So what does the host cell provide? Remember, DNA replication, you always need at least 1 protein, sometimes more, and that's why it's delayed. Viruses with small genomes need a lot of host proteins. They typically, as you will see for SV40, encode only 1 protein that attracts the host DNA machinery. But viruses with larger genomes, they encode all of the proteins needed for DNA synthesis and the production of dNTP substrate. So that's another thing you need, dNTPs, right? There are enzymes in the cell that make those, and these viruses with big genomes encode those enzymes as well. The polymerase, where does that come from? The small viruses with small genomes don't encode a polymerase. They encode a protein that, as I say here, orchestrates the host. You'll see what that means in a moment. The papillomaviruses, polyomaviruses, parvo, small genomes, they don't encode a DNA polymerase. But the large DNA viruses, as I said, encode either all or most of the replication systems, herpes viruses, adenoviruses, and pox viruses. And they can control it that way. Other proteins involved in DNA replication besides the DNA polymerase and some accessory proteins. There's an origin binding protein. There's always a protein that binds to the origin. We'll talk about that. There are helicases that separate the 2 strands. And there are exonucleases that are error-correcting enzymes. So this is something that RNA polymerases don't have. They don't have exonucleases that can correct errors. Remember, errors always occur. There are always misincorporation of bases. And so for DNA polymerases, they have exonucleases that can detect those and excise them so they can be fixed. And then that lowers the error rate for DNA viruses. And then the enzymes that make the triphosphates, thymidine kinase, ribonucleotide reductase, DUTPase, etc. So a lot of proteins involved in DNA synthesis. What statement about viral DNA synthesis is not correct? Large DNA viruses encode many proteins involved in DNA synthesis. Small DNA viruses encode at least 1 protein involved. Viral DNA replication is usually delayed after infection because it requires the synthesis of at least 1 protein and a viral protein. And all DNA polymerases are primer dependent, which is wrong. Let's see how we did. The answer is D. All DNA polymerases are primer dependent. That's wrong. For some of you picked the other ones. Large DNA viruses encode many proteins. Yeah, that's true, right? Isn't it true? All small DNA encode at least 1 protein. That's true. DNA is always delayed. Now, so those of you who pick those, they're individually correct, but I want to know which one's wrong. So it's all DNA polymerases are primer dependent. DNAs look different, right? And we've talked about this already. We have AAV parvoviruses with linear DNAs, and these have strange terminal repeats at the end. We'll see today what they're for. We have circular double-stranded DNAs. These are the small ones, 5,000 bases, don't encode a polymerase. Adenovirus, bigger, encodes the polymerase. Herpes and vaccinia, linear double-stranded DNAs that encode polymerases. So how do you accommodate all of these diverse structures in DNA? Well, we're going to talk about that today. So first is that you need to know there are two mechanisms of DNA synthesis. There's a mechanism called the replication fork and one called strand displacement, right? On the left, the replication fork is done by these viruses here. And on the right, the strand displacement is done by adeno and parvoviruses. So the replication fork is named so because it looks like a fork. You know, when you get to a fork in the road, take it. Do you know that quote? That's a fork, and you have DNA synthesis occurring on both strands at the same time. Okay, bidirectional DNA synthesis. On the right, the strand displacement is not really a fork because only one strand is being replicated. What happens here is that you have one strand being copied and it displaces the other strand, denatures it, removes it from the double strand. That's why it's called strand displacement. Now the replication fork is primed by RNA primers. That's what these little green things are here. The strand displacement is never RNA primed. It is either protein primed or DNA primed. So all the viruses we talk about can be put into one of these two mechanisms, replication fork or strand displacement, right? Now a big issue in DNA replication, and we'll solve it, is the 5' end problem. If you have a linear double-stranded DNA and you need primers to copy it, you end up with two DNAs after you prime and copy both strands. And the DNAs here are shown in red and orange, and they have an RNA primer still attached to them. The RNA primer is put down first, and then the DNA is made using the RNA as a primer. Then the RNA primer is removed because we don't want RNA in our DNA. And now we have a gap at the end that we can't fix, right? Because the only way you can fix it is by putting RNA in, and you'll remove it and get a gap. So this is the 5' end problem. And we're going to solve it today for multiple viruses. All right, so as long as you see this happens with linear DNA. So much of what we know about DNA synthesis came from studies of SV40, this small double-stranded circular DNA containing virus. Initially discovered as a contaminant of polio vaccines, and then it turned out to be an incredible model system. So the DNA is circular double-stranded DNA, about 5,000 bases, and it has a single origin of replication. That's where DNA synthesis begins, at the origin. And in this molecule, DNA synthesis occurs bidirectionally, away from the origin in both directions on both strands. All right? And that's illustrated here. So you have an origin. We've linearized the DNA just for clarity. We have an origin where DNA synthesis begins. The DNA is denatured, and now we're making already complementary strands in red, and that's happening on both strands. And then as these products get longer and longer, you can see you make a replication fork. So this is a replication fork-based mechanism. And this looks like a bubble in the electron microscope. And so this was a key experiment at the top here that showed that you got this bubble, but also it was bidirectional because they took SV40 DNA and they cut it with a restriction enzyme so that the origin was offset. It wasn't in the middle, right? It was towards one end, as you can see here. And then they let it undergo DNA synthesis, and the bubble got bigger and bigger, and it approached the left end, so showing that the replication is bidirectional. If they had put if they had cut it so that left the origin in the middle, the bubble would get bigger and bigger, but you wouldn't know if it was bidirectional. Right? So this experiment showed it's bidirectional and it occurs by a replication fork. So actually, this was the first information on DNA synthesis because we couldn't do this with eukaryotic cells. The DNA is too big and nobody had identified origins, but this little 5 kb genome, you could purify virus and you get pure DNA and you can use it for these studies. It's just fabulous. Okay, so this is a summary of what's going on here. Semi-discontinuous DNA synthesis from a bidirectional origin. So bidirectional origin just means that we're going in both direction. What does semi-discontinuous means? That means part of it is continuous and part of it is discontinuous, okay? The continuous part are the leading strands, which means that the polymerase, you could put down an RNA primer at the origin, and then the polymerase can start adding DNA and not stop because it's the right direction. It's 5 to 3 prime. And as the bubble gets bigger, the polymerase proceeds, and that product gets bigger and bigger. So 2 leading strands. The problem is there are lagging strands. This part, you cannot synthesize from left to right because it's the wrong direction. You have to go from right to left, which is 5 prime to 3 prime. But if you shrink this bubble, you can see you're only going to be able to make a short product primed with RNA. And as the bubble gets bigger, you make another short product. And as the bubble gets bigger, you make another one. It's the only way it works. That's discontinuous. So 2 lagging strands, 2 leading strands. Eventually, the RNA has to be removed and the gaps are filled in because now you have a DNA primer that can be used for that. And so you get 2 continuous strands. But in the initial synthesis, you get a leading and lagging. There is no end problem here because there are no ends on a circle, right? You can go all around the circle, make DNA, and it's complete without any problem. Those RNAs can be removed, and then the DNA in front of it can be used to prime, and in a circle, it's not an issue. So that's one solution to the end problem, to have a circular genome. Now, unfortunately, we don't have circular genomes, so we have other solutions to this issue. So let me show you the origin of SV40. Here, here's the genome, 5,000 bases or so. And there is base 1 and 5,243. We have early promoter here. And there's the origin, the core origin here. It's these repeated sequences in yellow here. So that comprises part of the origin. Actually, these elements are all part of the origin as well. And you see LT binding sites here. That's large T antigen, which plays a big role in DNA synthesis. That is the one viral protein that has to be made in order for DNA synthesis to occur. So you can see the origin overlaps the early promoter. And if you go a little to the right, you find the late promoter as well. So I want to show you how this works, just so you have a mechanistic view. You don't have to memorize any of the components. The only thing you need to know is T antigen, because it's so important. We're going to talk about it for the rest of this course in other functions. So there's the SV40 DNA at the top. There's the origin of replication. And the origin is rich in ATs, so that makes it easy to denature, right? If you had a lot of GCs, it would be hard. That would not make sense. And then you have a large T binding site. So what happens is when this DNA gets in the cell, the early promoter is active. It makes an mRNA encoding large T antigen. And when you get enough large T made, they bind to the origin. Right? So that's the one protein that you need to make in terms of SV40. And you can see 12 large T molecules bind the origin, 6 on either side. All right, and so then there's an ATP dependent reaction that helps to denature the template. So T antigen is a helicase, which denatures the DNA. And it also allows this protein, RPA, to come in. RPA binds large T, and that transfers it to the single-stranded DNA. So this binding keeps the template single-stranded. So large T comes in, denatures the double strand, it recruits RPA, which then binds the DNA and keeps the single strand separate. So that's the function of large T. Can you imagine if this DNA went in the nucleus and just waited for the cell to replicate it? Would never happen. One little circle in the genome of 3.2 billion bases wouldn't happen. So you need to have LT to attract the cell system, because that's what's going to do everything else. All the other proteins involved in replication of this viral DNA are coming from the host cell. Okay, so then what happens is a primase comes in, and again, it's recruited by RPA and LT. It's called Pol alpha primase, and it makes the RNA primers that are needed for DNA synthesis, and it also makes short DNA fragments. So that's shown here. We have green RNA primers being made and short DNA fragments. Okay, and then we make longer DNA by a second protein called polymerase epsilon, which comes in and makes the longer leading strands and the lagging strands as well. There are also other proteins that come in here and are involved with keeping the strands separated, but we don't need to talk about that. All right, so primase makes the primers, short DNAs, then epsilon comes in and makes longer DNAs. And large T remains bound so it can continue to denature the DNA. And at this point, you're making long DNA. And that gets longer and longer. You can see our leading strands and our lagging strands here. At some point, the RNA is removed by RNase H. The gaps are filled in, and then DNA ligase ligates the DNA. So now we have double-stranded DNA, which has no RNA in it, and it's just continuous strands. There are no gaps or anything inside it. So that's how you replicate the genome. And you go all around the circle that way. This is a diagram of the polymerase and how it works. There are actually some great movies on YouTube that illustrate this. Here we have large T hexamer, and then we're showing the primase here, the RNA primer, and the polymerase epsilon that is making the long DNA. So there's a template. It's being denatured by LT, and then the two strands are being copied at the same time, right? Semi-discontinuous replication. Now, when you get to the end, you take the template and you copy it, you end up with this. You get two molecules, but you can't separate them. They're like this. So that's where topoisomerase comes in. It nicks both strands of one molecule and then religates them. So now you have two strands. Topoisomerase 2, that's its function, to be able to, what's called resolution of the two strands. Otherwise, they'd stay linked together. That would not be good. All right, topoisomerase 2. Now, topo 1 works during replication because as you are making DNA, this closed circle gets all wound up like a rubber band, right? As the two top strands are denatured and you synthesize, the bottom is getting wound up. So the topoisomerase 1 nicks one strand and it relaxes. Can you imagine that, looking at that in the cell, boom, and the thing all relaxes to this, which is called a relaxed supercoil, and then it goes on and it gets all wound up and it nicks again. So topo 1 and 2 have essential roles in DNA synthesis. These are cellular proteins, okay? They are not viral proteins. Okay, next question. The SV40 genome is a circular double-stranded DNA. Which statement about its replication is correct? A, viral T antigen binds and unwinds the ORI. 2, replication is bidirectional from a single ORI. 3, the 5' end problem is solved. D, it has leading and lagging strand synthesis. All of the above. Which is right? Okay, how did we do? Yep, all of the above. The T antigen binds and unwinds the ORI. Replication is bidirectional from a single ORI. The 5' end problem is solved, and it has leading and lagging strands. All of the above. are correct. Okay, so that was SV40, double-stranded circular DNA. Let's look at parvoviruses that have a single-stranded genome with these structures at the end called terminal repeats. And at the bottom is the sequence of the terminal repeat. So basically, most of the genome is single-stranded. It encodes proteins involved in DNA synthesis and capsid proteins. But the sequence of this terminal repeat is shown here, so you can see it forms base pairs. So this sequence base pairs, and it forms this base paired structure at the end, ending in this 3' hydroxyl. This is a perfect primer for DNA synthesis. It's built into the genome. You don't need to go anywhere else. You don't need to make RNA. And that's what it is. It's the primer for DNA synthesis, and the primer is a DNA. And here's how it works. It's a little complicated, but I'm going to go through it, but then you could sit down and look at it and get it. So there's the genome. And the inverted terminal repeats we have labeled ABC, A'D, and the A and the A' are complementary, okay? So this forms a hairpin, as you can see here. I showed you in the previous slide. Now, this gets into the cell. This is what's packaged in the virus particle. And when it gets in the cell nucleus, the cell recognizes it as broken. So it fixes it, it repairs it. So cellular polymerase delta fills in that gap because it does not like single-stranded DNA. It wants to make it double-stranded, so it does. And now you have a double-stranded genome. That can be transcribed now. Before you couldn't transcribe it. And you can make some proteins that you need for DNA synthesis. And one of them is called Rep 7868. And what that does, it makes a nick, a sequence-specific nick between A and D in this terminal hairpin. All right, so then you end up with this molecule, which is pretty much the same, except it's got a nick. It's no longer continuously double-stranded. Then the polymerase elongates from the nick, from D to the left. And so now you're basically copying that blue hairpin, and you have a red product. So now you have a fully double-stranded copy of the genome, including both terminal hairpins, which is what you need, of course, in order to make more of it. So that's the replication intermediate. It then forms hairpins. They spontaneously form, and the 3' end can serve as a primer for strand displacement. That's what's happening here. So the red terminal repeat, right, it is got a 3' hydroxyl from the extension, and that's going to serve as a primer for strand displacement DNA synthesis. So the top strand is displaced. So you end up with the top strand here, right, which is single-stranded, and the product, which is the double-stranded product made by copying the blue strand. And the blue strand and the orange strand are shown there. And that will re-enter the replicative cycle, get nicked, and go through all of this again. So continuous replication, there's no discontinuous replication. It uses its inverted terminal repeat to self-prime. It uses these enzymes from the host cell. Rep 6878 are needed for nicking the double-stranded DNA, and this is all by strand displacement. It's no replication for just strand displacement. But you can see because there is a DNA primer, there's no end problem. These terminal repeats ensure that you can copy all the way to the end without a problem. So here, this molecule is a complete copy of the DNA, and then all you need to do is nick it. Sorry, it's already been nicked and copied, but all you need to do is let it self-prime, and no sequence will be lost there because you're using the genome as a primer. Really good solution to the end problem. This is done by single-stranded viruses all over. All right, getting bigger, adenovirus. Here we're going to see protein priming. The DNA is linear double-stranded, about 36,000 base pairs, and there's an inverted terminal repeat at each end. And then the protein primer, it's called TP, terminal primer, terminal protein, sorry. And it's attached to the DNA because it's been used as a primer. We'll see that happen. So there's an origin at both ends because that's where DNA synthesis begins, at both ends. So that's what an origin is, the place where DNA synthesis begins. This is going to be strand displacement, as you'll see. Okay, here is adenovirus, here, the DNA. And we're looking at the synthesis of the primer here. So the polymerase here in purple is bound to the pre-terminal protein. And the polymerase adds a C to it, a cytidine, okay? And this is added to a serine residue of the terminal protein. That C is going to base pair with the G in the genome. This is the 3' end of the viral genome. Here's a pulled out view of it. And that's going to serve as a primer, and the polymerase is going to start adding bases, as you can see here. So it's adding C18. It actually slips back a couple of bases because it starts priming internally, and then it slips back, and it's okay because the last 6 bases are repeated, GTA, GTA. So the cat binds to the GTA, and then the polymerase goes and copies the rest of the genome. And in so doing, it detaches from the pre-terminal protein. So now the pre-terminal protein is left bound to that first 5' base C on the viral genome. Okay, so that is illustrated here. Now we're going to take an overview of what is happening. So this is strand displacement, right? The polymerase is copying the light blue strand. It's displacing the top strand. And in step 2, we see the red product complementary to the light blue strand. Top strand is being displaced. And as it's displaced, a viral protein is binding to it, that yellow protein. It's the DNA binding protein to keep it single-stranded. And in fact, once you get the whole top strand displaced, it's coated in single-stranded DNA binding protein, with the exception of the inverted terminal repeats. So because they're inverted terminal repeats, they can base pair. And they're not occluded by the DNA binding protein. And guess what? They look just like the end of the viral genome. And so the polymerase will then come in and start copying this strand by the same protein priming mechanism. So that's the top strand, which was displaced. Meanwhile, we have a double strand product made by the initial copying. So we're going to end up with 2 double-stranded products. This is semi-discontinuous replication, okay? No end problem because of this. The polymerase is able to prime a few bases internally, then slip back, and then extend. So there's no need, and the protein primer helps with that. So there's no need for an RNA. There's no end problem. And it's strand displacement. The DNA polymerase, the DNA binding protein, these are all viral proteins doing this. So an example of protein priming. We saw another example of protein priming for poliovirus RNA synthesis. Here's a picture of the adenovirus single-stranded DNA binding protein. That's the actual structure there. It's been likened to a plow, right? This little plow-like protuberance, and the idea is that as the polymerase is moving along the DNA, denaturing the parental DNA, right? It's strand displacement. This is the displaced strand on the top. These single-stranded DNAs are binding, and perhaps this little protuberance helps to denature the double-stranded parental DNA. So, next question. How is DNA replication of parvovirus and adenovirus similar? So parvovirus was the single-stranded DNA virus we looked at. How are they similar? They both require protein-linked primers. Replication occurs by strand displacement. DNA synthesis occurs in the cytoplasm. The replication fork occurs in both none of the above. How is they similar? I'll tell you, I didn't tell you this. Most DNA viruses go to the nucleus to replicate. The exceptions are viruses that stay in the cytoplasm, like poxvirus and giant viruses, but none of these are giant viruses. Okay, let's see how we did. Yes, replication occurs by strand displacement. That's what they have in common, right? Parvovirus, strand displacement, adenovirus. They both require protein linked. Adenovirus does, but not parvo. It's a DNA primer built in the genome. DNA synthesis occurs in the cytoplasm. I didn't tell you that, but for both viruses, it occurs in the nucleus. A replication fork occurs in both. There's no replication fork in either. It's strand displacement. So that's why it is B. So again, let me emphasize, because I haven't made that clear. Most DNA viruses replicate in the nucleus. Parvoviruses, SV40, adenovirus, herpes virus. The one exception we'll talk about briefly is vaccinia virus, a poxvirus, which sets up shop in the cytoplasm, so it cannot use nuclear proteins, polymerase and so forth, so it makes its own. But that's in the cytoplasm. Everything else is pretty much in the nucleus because that's where all the machinery is. All right, herpes simplex virus getting bigger. Now the genome is over 100,000 bases long, double-stranded DNA, and it has 3 origins of replication, ORI L, S, and S, 2 ORI S's. There are a lot of repeated sequences. These are called TRL, IRS, and so forth. And it encodes all these proteins that are needed for DNA synthesis. Primases to synthesize the primers. Processivity protein, which means it helps the polymerase to keep going and not fall off. Origin binding protein, single-stranded DNA binding protein, and there's the RNA polymerase, the DNA polymerase. The DNA enters as a linear molecule, so it's in the particle as a linear molecule. It enters the nucleus, and then it's converted to a circle and replicates by rolling circle mechanism, which is we're going to talk about. So there's the genome of herpes viruses. There's a cellular ligase with that name that converts it to a circle. So the DNA, the linear DNA is converted to a circle. So host proteins are responsible for circularization. And this is rolling circle replication. One year, one of the students in this class likened it to toilet paper, right? Just pulling toilet paper off of a roll. The roll keeps turning, and you get the toilet, and each sheet could be a genome, right? Think of it that way. Maybe it helps, because here we have our double-stranded circle. It's nicked by an endonuclease, so now we have a 3' hydroxyl, which can serve as a primer, all right? So there, we can do that, and we can start making DNA right on that, so we don't need any RNA primers for that anyway. And so the red is the newly synthesized DNA, so it is displacing the top strand, and eventually the red DNA will cover the entire genome, and as it gets longer and longer, the other strand is displaced to a greater extent. You can see that here, the blue strand. And once it's a certain length, then you can have discontinuous DNA synthesis happening on the displaced strand. So continuous DNA synthesis around the circle, then discontinuous, these are primed with RNA, and then DNA is made, then the RNA is removed, and the gaps are filled in. And these molecules get very long. This keeps going. It's like you keep pulling the toilet paper. It just keeps coming out. This goes on, and you make multiple genome length molecules. Dozens and dozens of genomes or more are part of these molecules. And each one doesn't have an end problem, right? Because the DNA polymerase can go all the way to the 5' end, and then at the 3' end, if there's an RNA primer, it can be removed, and then the preceding DNA can be used to fill it in. The only end problem would maybe be the very last genome. If this circle fell apart and you were left with the double-stranded DNA, maybe you'd have one end problem, but in general, there's no end problem here because of the rolling circle replication. All right, so displacement is continuous DNA synthesis, and then the discontinuous occurs on the displaced strand. No end problem, and the primers are only used here because, as you can see, there's no opportunity to have a DNA primer, whereas there was here by making this 3' hydroxyl. There's no opportunity for a DNA primer, so we need to use RNAs. So genome length means we've copied this circle, and now we have pieces that are equivalent to the whole genome, which are going to end up being packaged in the virus particle. So this is another solution to the end problem. And finally, pox virus. So this is the one virus that we talk about that replicates in the cytoplasm. So it encodes all proteins needed for DNA replication. It also encodes a DNA-dependent RNA polymerase, because all that stuff is in the nucleus, and apparently this virus cannot access it. It makes, in the cytoplasm, what are called factories, and it sets up shop there. And these are the factories illustrated in this experiment. So this is a cell infected with pox virus, with a pox virus like vaccinia virus. So pox viruses include vaccinia virus, monkeypox virus, and smallpox virus. And people work in the lab on vaccinia because smallpox is too dangerous and it's been eradicated, so you can't work on it. And vaccinia virus is relatively safe. So here is an infected cell. It's stained with DAPI. The blue stains DNA, right? DAPI stains DNA. So you can see the nucleus there. And then there's some DNA in the cytoplasm. That's viral DNA being replicated in the cytoplasm. So these are what are called factories, where viral replication occurs in the cytoplasm. The second frame, it has been stained with an antibody to the DNA binding protein. So just like adenoviruses, pox viruses encode a DNA binding protein that's used for DNA replication. That's a viral protein. You can make antibodies to it, and you can see the DNA binding protein is in these cytoplasmic factories. It's not in the nucleus, so DNA synthesis is occurring in the cytoplasm. And that's a merge of the two colors, which emphasizes that the DNA binding protein, which marks the sites of poxvirus replication, is in the cytoplasm, okay? So this is all cytoplasmic, and all the proteins involved are viral. And at least 15 viral proteins are involved in DNA synthesis. This is a model for how it occurs. The vaccinia genome is interesting. It's double-stranded DNA, so it's base paired along the way, but the ends are covalently linked. So it's not really a circle. It's just a double-stranded DNA with the ends covalently linked. And what happens is that replication initiates at the ends. This is probably an origin of replication. There are primers, RNA primers put down to initiate, and then DNA synthesis occurs continuously all the way around the circle. So you get a dimer, and these can be separated probably by topoisomerases. But the key here is that when the DNA comes all the way around, these RNAs can be removed, the DNA can be filled in. There's no end problem. Why is that? Because this acts sort of like a circle. Even though it isn't a circle, it's just it's a fully base paired DNA molecule. It acts like a circle, and there's no end problem. Last question. What makes poxvirus DNA different from all the other viruses we discussed? The DNA polymerase requires RNA primers. DNA synthesis occurs in the cytoplasm. DNA synthesis occurs by strand displacement, or none of the above. What makes it different? Okay, how did we do? DNA synthesis occurs in the cytoplasm. That's how it's different. It requires primers, but so do some of the other viruses. There's no strand displacement. So it's B. All right, let's talk very briefly about origins of replication. We've mentioned them in terms of SV40, which has one origin. Adenovirus has one at each end. Herpes virus has three. What are they? These are segments recognized by viral origin recognition proteins. They're typically AT-rich to facilitate their denaturation so that the polymerase can get at both strands. And that is where the replication machines assemble. And as you've seen, some have one and some have multiple origins. So here are origins of SV40, HSV, one of the HSV1 origins, and adenovirus 5. So here, for example, on SV40, there's the core origin. There's the large T binding site. AT-rich sequences are shown in green. So again, that facilitates denaturation. The yellow are sequences bound by the origin recognition proteins here, SV40, herpes, and adenovirus. It's right at the end. The origin recognition protein is the polymerase and terminal protein together. And there also happen to be binding sites for transcriptional regulators sprinkled throughout these origins. They're always very close to each other. And you can see here in the herpes ori that there are also two promoters going in opposite directions here, indicated by the red arrow. And NF1, OCT1, these are transcriptional regulators. So origins and transcriptional regulators always near each other. And then the viral origin recognition proteins, we've mentioned them, but let's summarize. Polyoma T binds DNA, right? It binds, here's origin binding proteins. Viral polyoma T, the red, binds the origin and recruits cellular replication proteins. The parvovirus Rep6878 binds at the ends. It unwinds the DNA, something I haven't told you, but it's also needed for the nicking of the double strand to make a primer. The adenovirus pre-terminal protein binds at the end and recruits the polymerase. So technically, the pre-terminal protein is the origin binding protein, and it recruits the DNA polymerase, which is a viral protein. And finally, the herpes virus UL9 protein is an origin binding protein, which recruits viral proteins to the origin to do replication. So it's interesting that these origin binding proteins, one of their jobs is to recruit the DNA synthesis machinery. For the small viruses, that's cellular. But for the big viruses, it's viral. So even if you encode your own proteins to make DNA, you have to recruit them to the DNA, and you use origin binding proteins to do that. Now we're going to come back to large T in a couple of lectures. So I want to show you this protein. This is the most studied protein on the planet. Every amino acid has been changed, and the properties of the protein have been studied. So there's the protein at the top. It is 708 amino acids long. And some of the features of the protein are shown here. We'll go through a few of them. So for example, this is the part of the protein that binds the origin. Here's the nuclear localization signal. Here's the part, here are parts that bind polymerase alpha, here and here. And this is very interesting. This is a part that binds RB. Remember retinoblastoma protein? We're going to come back to that. So T is a species-specific DNA binding protein. What does that mean? It means the SV40, which is a primate virus or T antigen, will not bind the origin of a mouse polyoma virus. So these pre-initiation complexes referring to the origin ori complexes don't form in the wrong species, and they do not interact with the cellular primary, so you don't get replication. This protein also binds and sequesters cell cycle regulators. That's what RB is. It's a cell cycle regulator. And in fact, T antigen makes cells enter S phase. This is very important. What does that mean? So most of the cells in your body do not divide, right? There's no need for them. The exceptions are the mucosal epithelial cells, which divide. very quickly and turn over in a week or so, but most of your other cells are not dividing. Viruses need to have replicating cells in order to reproduce. They don't do well in quiescent cells. And so a virus floating around in your body, how will it detect a cell that's dividing? It cannot. You can't tell from the outside a dividing cell. It looks like every other cell. So what they do is if they get in a cell and it's not dividing, they induce it to divide. How do you make a cell divide? You turn on DNA synthesis. That's the key. You turn on the DNA replication machinery, which is quiescent in most of our cells. You turn on DNA replication, and that will drive the cells eventually to divide, because you'll have 2 times the number of DNA molecules that you need. The cell knows that and will split and give one cell one complement and one the other. So viruses induce DNA replication, and that's done by early or immediate early gene products. It has to be done early, as soon as the virus gets in, right? Otherwise, it's going to be too late. So this is how this happens. Here's the cell cycle, right? There is a roughly 24-hour cell cycle in most cells, where there's mitosis is when the cell becomes 2, right? So it has gone through replication of DNA and growth. So the cell gets bigger, it replicates its DNA. Now it's basically 2 cells in one, and it splits in half. And these are normal-sized cells with the right number of chromosomes. So that's mitosis there in red. Then you have a period of cell growth, you have replication of DNA, another period of growth, and then mitosis. So here's retinoblastoma protein. It is a restriction point. It says, do I really want this cell to divide? Are conditions right, or am I being fooled? So it can sense that, and it can stop the cell cycle. RB protein can stop the cell cycle in its tracks, and the cell will never divide. We'll never get to replication of DNA, because that's downstream of RB. So RB controls entry into S. How did we discover RB? Kids have retinal tumors up to about 5 years of age. They have retinal tumors where RB gene is deleted. So you take out RB. Now there's no checkpoint in the retinal cells. They just keep dividing. And when cells divide uncontrollably, that's where cancer comes from. Because at each cell division, the cell accumulates mutations. And eventually you get mutations in about a dozen proteins, including oncoprotein genes, and that's it. Then you become a cancer. So these kids lost their RB gene, and as a consequence, eventually developed retinal tumors. And it was figured out, and they said, oh, there's this protein, RB. And then it was studied more, and turns out viruses interact with it. Why? Because it stops the cell cycle, so viruses want to antagonize that, right? They want to antagonize RB, and you've already seen how this happens. And I said, remember this. So the adenovirus early promoter needs a cell transcription protein called E2F. Normally E2F is bound by RB, so AD can't get it. So adenovirus makes E1A, and it binds RB and frees up E2F. Turns out that SV40 large T and human papillomavirus's E7 protein also bind RB to antagonize it. Why do they do this? When you bind RB, that makes the cell divide, right? Because you no longer have this checkpoint. So SV40 and the papillomaviruses, and to some extent adenovirus, they bind RB, and that gets the cell dividing. There are genes that encode DNA synthesis proteins of the cell and everything you need to divide, and those are controlled by E2F. And normally, RB is bound to that, and so you don't get activation of those genes. That's how it acts as a checkpoint. But if you have viral proteins antagonizing RB, you will now be able to activate those promoters, which are E2F dependent, and they will make proteins that are needed for DNA synthesis and for passage through the cell cycle. So that's how RB works. It represses the genes you need for cell division. And the viruses counter it by binding RB. And that allows the cells to start dividing, and it provides a good environment for viral DNA replication because the cells start to make DNA, and they make all the proteins that you need for DNA synthesis, and the virus can now use them in the nucleus. Because if the cell is not dividing, it's not making DNA polymerase or anything else that you need for DNA synthesis, okay? You can imagine that this is dangerous. If things get out of control, the cell could divide forever. And what did I just tell you? That's a recipe for cancer. So we're going to see in a couple of lectures that actually does happen. So next time, Monday, we're going to talk about reverse transcription and integration. This is one of my favorite lectures, my second favorite. You will find out why some chicken eggs are blue.